Thank you for inviting and providing an opportunity to talk this excellent webinar. My name is Taketoshi Kodama. I'm from the University of Tokyo, and today's my talk is about how the shrinking process is different among 5 plankton groups. We had submitted this topic as a preprint, received reviewers' comments, and now we are revising the manuscript. So if you have questions and comments, please ask me during the webinar or please email me. This is my email address. So let's start with the introduction. As we know, phytoplankton significantly contributes to the carbon dioxide removal, and the phytoplankton community and biomass in the surface ocean impacts the biological carbon pump. The left figure is from Honjo 1982, and he reported that POC flux in the deep ocean increases soon after phytoplankton blooms. The first one is after the phytoplankton diatom blooms, and the second one is after the unusual Cocollosphere bloom. Some studies pointed out chlorophyll A and its breakdown products, such as pigments, are richly contained in the sinking particles. Also, large diatom dominance in the surface layer possibly enhances the POC flux in the ocean because the increase of POC flux is observed after spring bloom, and POC flux is high in the productive diatom abundant ocean. However, recent development in molecular biological techniques show that small phytoplankton Synechoccus plays a significant role in POC flux, especially in the oligotrophic ocean. This figure is from Guidi's Nature paper, which shows Synechoccus are important for increased POC flux as well as heterotrophic dinoflagellate Noctiluca scintillant. So now we need to evaluate the phytoplankton community in the sinking particles to understand their role in the biological carbon pump. Also, we must care about this ungrazed written by Boyd and Newton, because zooplankton grazing is another significant role in the biological carbon pump. Numerous studies pointed out that zooplankton faecal plates significantly contribute to POC flux. The faecal plates are large and heavy, so they sink faster than phytoplankton cells. Of course, the faecal plates are digested materials, so the phytoplankton cells are digested largely. However, a part of phytoplankton cells are not digested, for example, the resting spore of diatoms. This figure is from Kuwata and Tsuda. They fed diatom kit cells, resting spore to compose neocorns, and reported most of the cells in the faecal plates were undigested and alive. Moreover, Wilson and Steinberg observed numerous fresh Synechoccus in the compost gut contents. So we have a hypothesis that healthy and fresh phytoplankton cells are provided to deeper layers selectively by zooplankton grazing. Our motivation is to understand how prey-predator relationships change the biological carbon pump in the ocean. As the first step, we start our study in order to know which phytoplankton is transported to the bathypelagic layer by the grazing processes. Now we selected the Sea of Japan as a sampling site. We consider this sea as an ideal sampling site. First, the surface temperature, sea surface temperature of this area is rapidly warming. The warming speed is the fastest in the global ocean. Not only the surface temperature, but also the warming trend is observed from the surface to the deeper layer. Because this sea is semi-closed sea with its own deep convection system, this warming trend slows down the deep convection and decreases the oxygen concentration in the middle and deeper layer. This figure is from Chen's Nature Climate Change paper, and the AOU, apparent oxygen utilization, was linearly increased. On the other hand, plankton ecosystem of the Sea of Japan is very similar to those of the North Pacific. The white plankton community shows that seasonal variations and diatoms, the orange one, are high in the winter to spring. And Prochlorococcus, the green one, and Synechococcus, the light blue one, are high in the summer and autumn. Also, there is a Bathypelagic Bendicularian in the sea. This kind of zooplankton is known as an essential player in the biological carbon pump because it repacks the suspended particles in the mesopelagic layer and enhances the biological pump. These rapid changing physical and chemical environments and typical plankton ecosystems in the North Pacific indicated that the Sea of Japan is an oceanic microcosm for biological carbon pump studies. So we moored to sediment traps at Station Fato in the Sea of Japan. The setting depths were 387 meters and 990 meter depths, and the moor period was from 22nd of June 2022, 25th of May 2023. Now I call 387 meter depths as 400 meter depths and 890 meter depths as 900 meter. The sampling bottles were changed every 26 days, so we collected the sediment into 13 bottles at each depth. This is a photo of this sampling bottle. At 400 meter depth, the third bottle was overflowed by unfilled, and from host to the end, the sediment were not observed in the bottle. So we used only first two bottles to understand the vertical difference of phytoplankton composition. The environments during the moor period are like this. The blue, red, green lines indicated sea surface temperature, mixed layer depth, and sea surface chlorophyll concentration respectively. When we focused the first two bottle period, the environmental conditions were relatively stable. The chlorophyll air concentration was low, MLD was shallow, and SST surface temperature was relatively high. Next, I introduced the sampling treatment. All of the samples were preserved in the 5% formalin neutral buffer solution. Before analysis, we removed swimmers from the bottles and filled the bottle with the formalin solution again. Then we took 5 ml of solutions with particles, and the particles over 0.2 micrometer were collected on the membrane filter. The inorganic carbon particles were removed by hydrochloric acid, and weights and carbon contents were measured. The other 1 ml solution with particles were observed under the microscope for fecal plates analysis. The fecal plates was classified into 4 types. That is, ellipsoidal, cylindrical, tabular, and spherical types. All of the fecal plates' lengths and width were measured, and their carbon mass was calculated. The phytoplankton in the sediment traps were identified by amplicon sequence. We prepared 6 types of subsamples from the sediment trap bottle. One is complete sample, which can show the whole phytoplankton community in the sinking particles. The faker pellets were prepared 4 types: a single ellipsoidal pellet, may originate appendicularians, and a single cylindrical type may originate from euphagites, 10 spherical may originate from small copepods, and a single tabular faker pellets may originate from salps, but very small compared to the previous studies. The samples after removing all faker pellets are treated as aggregates. In addition, suspended particles in the subsurface layer was investigated. The DNA extraction was conducted using CAMP DNA FFPE tissue kit except for suspended particles. The difference from the conventional method is a heat step. The samples were incubated 19 degrees for 1 hour to reverse formaldehyde modification of DNA. If we cut this process, the extracted DNA cannot be amplified with the following PCR step, but this process fragmented the DNA. Amplicon sequence was conducted for 16S ribosome RNA gene before V5 region. This region is well known as metabarcoding analysis to prokaryote community, but the chloroplast genome in the eukaryotic phytoplankton can be amplified as well. However, the dinoflagellate chloroplast genome evolved ultra rapidly, and so it cannot amplify this primary set for this region. The 80S RNA gene amplicon sequence could amplify the dinoflagellate genome, but phylogenetic resolution is not high at this region, so we did not treat dinoflagellate in this study. The sequence results are analyzed by QIIME2, and ASVs were identified based on Silver and PR2 databases. First, we check the POC flux. The flux at around 400 meter depth was same with around 900 meter depth. We did not conclude why this occurred, but it may be due to the active flux of zooplankton or micronecton, or low sampling efficiency in the upper sediment traps. The faker pellet contribution was low but comparable to the other areas. The contribution was around 4% in the upper layer, while it was 7 or 8% at 900 meter depth. When we see the breakdown of the faker pellets, ellipsoidal type are dominant in every bottle. The difference in faker pellet flux between two depths is due to the contribution of ellipsoidal pellets. The increase of ellipsoidal faker pellet indicated that the packing of suspended particles occurs between these two depths. When we check the size of the ellipsoidal faker pellet, the size was significantly larger in the deep than the upper one. As I introduced, the mesopelagic appendicularians are present in this sea. Thus, we concluded that appendicularians repacks between these two depths. Then, what type of phytoplankton sinks? This figure showed the vertical difference in phytoplankton community structure based on amplicon sequence. Since we cannot find a significant difference between the periods, we show the mean values here. The far left bar is phytoplankton community at 50 meter depth, corresponding to the bottom of the euphotic layer, and the right four birds is the phytoplankton community in the sediment trough samples. At 50 meter depth, we can find that haptophyte, shown as a purple, were dominant. On the other hand, in the sediment trough samples, the contribution of haptophytes is very small at both 400 and 900 meter depths. So we consider that the haptophyte is easily decomposed during the sinking processes, and this group does not directly contribute to the POC flux. The second characteristic concerns the green or orange groups. These belongs to diatoms. The diatom contribution is low at 50 meter depth, but high in the sediment trough samples. Compared among the sediment trough samples, the diatom contribution was high in the aggregates than the complete samples, and high in the 400 meter depth than 900 meter depth. Instead of the diatoms, Synechoccus contribution was increased in the complete samples than and 900 meter depths. Let's see phytoplankton community in the fecal plates. This figure compared the aggregates and fecal plates at 400 meter depth. As we can see, kid cells diatoms, the dark green one, are dominant in every type of fecal plate. The other diatoms are high in the aggregates, but low in the fecal plates, except for Trachyocera of ellipsoidal pellets. When we see the result on 900 meter depth, Synechoccus contribution is high in the fecal plate. The contribution was over a half in the cylindrical plate. We also find Pelagomonas contribution was very high in the spherical plates, but it was a single experiment. So we are now trying to edit the spherical samples. As a result, we can see the kid cells and Synechoccus are important for POC flux through the zooplankton grading, but not diet except kid cells. However, we must care composition data is not biomass, and the fecal plate contribution is very low in the POC flux. So we used ASV richness, the number of species based on ASVs. First, we compared the plankton richness and POC content in a single fecal plate. Only the spherical plate is not a single one. The difference in the color and shapes indicated the fecal plate types, and the gray band denote the result of aggregates whose POC flux was 2 or 3 orders higher than a single fecal plate. We can find a positive relationship between ASV richness and POC content, suggesting that increase of POC flux increases the phytoplankton diversity and their biomass. Even though the POC content was 2 orders smaller than the aggregate samples, the phytoplankton diversity in the fecal plates are sometimes same level with the aggregates at 900 meter depth. This suggested that fecal plates contains phytoplankton abundantly in particular at 900 meter depths. The most interesting results are found in Shinnecoccus. The Shinnecoccus ASV richness was very low in the aggregates, while it was high in fecal plates. In the large plates, the Shinnecoccus ASV richness is higher than the aggregates. Also, the richness at 400 meter depths was lower than 900 meter depths. These indicated that the Shinnecoccus is concentrated into a fecal plate in the Bathypelagic layer. On the other hand, the sinking particles except fecal plates, Shinnecoccus was very rare. Next, we see the result of the kit cells. The positive relationships was observed, and the richness in the large fecal plates are same level with the aggregates. The depth difference was not obvious. These indicated that fecal plates transport kit cells effectively as well as Shinnecoccus, but sinking process without fecal plates can transport kit cells to the deeper layer. On the other hand, the diatom richness except kit cells is smaller in the fecal plates than the aggregates. This indicated that diatoms are not effectively transported by zooplankton grazing processes, and the sinking process without grazing is more important. Why are only kit cells and Shinnecoccus transported in the fecal plates? Kit cells form resting spores, and the spore can survive in the dark ocean and in the gut of zooplankton. We consider the formation of resting spore is important for the transport processes in diatom groups. However, it is known that tertiary diatoms also form resting spores, but their contribution is low throughout the sample. We don't know why, but our new results show that tertiary increase in the winter time, so there is a seasonality in the diatom composition. On the other hand, how the Shinnecoccus survival from the grazing was not clear so far. But the previous study by Gorski showed that living Shinnecoccus was observed the appendicularian fecal plate. So Shinnecoccus may have biological properties against the zooplankton grazing, and they contribute the downward carbon flux only when they are preyed by zooplankton. This is our conclusion. The difference of the surface community and the sediment trap samples indicated haptophytes are easily decomposed and diatoms are abundant in the sediment trap samples. The difference of the aggregates and the fecal plates indicated that kit cells and Shinnecoccus abundantly exist in fecal plates, and repacking process between 400 meter to 900 meter depths enhances Shinnecoccus sinking below 900 meter depths and then Bathypelagic layer. Thank you for listening.